The next phase that we see in our cell cycle after interphase, where we have our G1, our S, and our G2, if we remember those terms, is that we're going to go through mitosis. And mitosis is the division phase of the cell cycle. So this is when our one cell is going to become two identical daughter cells. So we can see in our picture here, we start with this one original cell, and as it goes through mitosis, we're going to get two identical cells to end with. So I'm briefly just going to show you all of our phases of mitosis before we talk about each of them individually. So you won't have anything to write down here. But first we have the prophase cycle. Prophase is the first phase of our mitosis. Then we have metaphase, and then anaphase, telophase, and then cytokinesis. Cytokinesis is separate from mitosis, but we will kind of lump it all together. But we are going to spend a bunch of time talking about mitosis, and I will often shorten their phases to just their first letter. So we will often shorten it to just the first letter, so we will refer to it as P-M-A-T, PMAT. So the first phase we have is P, so we're talking about prophase. And in prophase, our chromosomes are going to condense and are visible as sister chromatids. And if we remember, our sisters are identical copies. So they already went through replication during interphase, and so now we have two copies. So each half of this little one here are the same, each half of this white one are the same, making them sister chromatids. In addition to this, during prophase, our nuclear membrane is going to disappear. So our DNA is going to start spreading out. So this line we have here around outside of our nucleus is going to go away, giving us access to the DNA inside. And our spindle fibers, which is a new term, are going to form out of centrioles. So we've seen centrioles before. This is our organelles that help pull chromosomes apart during cell division. So our spindle fibers are these little structures here on these guys. They're going to reach out their little spider-like arms, hook onto the DNA, and pull them where we want them to go. The next phase in PMAT is our M, metaphase. And in metaphase, our spindle fibers are going to connect to the centromeres. The second thing that's happening in metaphase is that our chromosomes are lining up in the middle of the cell. So I like to remember metaphase by their meeting in the middle. So in metaphase, our chromosomes line up in the middle. They get moved there by the spindle fibers. They're going to pull them into place. The next phase we have in PMAT is anaphase. So this is the A portion of PMAT. And in anaphase, our sister chromatids are going to be separating and pulling away from each other and becoming individual chromosomes. So we can see that in our picture here, that we had our X's and now they've split in half, going to opposite ends of the cell, which is our next part. So our chromosomes are separating and being moved to opposite ends of the cell. The last phase we have of PMAT is our phase telophase. Our chromosomes are decondensing and starting to look like chromatin again. So chromatin is what our DNA is normally in, and it is this thread-like or rod-like. I like to think of it as a ribbon. So chromatin, ribbon, they rhyme, helps me remember it. So we can see DNA in mitosis and these nice fancy little rods all condensed together, but normally it's just a whole bunch of threads. And we're trying to go back to a normal cell, well, two normal cells. So our nuclear membrane reforms around chromosomes at each pole. So we can see in our picture here that we have two nuclei back in our cells. So we're trying to get to two separate cells now and back to normal cells. And our spindle fibers are going to break down. The only thing we use them for is cell division, so we don't need them anymore, so we're going to get rid of them. And our process of cytokinesis is going to begin during telophase, which is why we kind of lump it together into mitosis. All right, and lastly, we have cytokinesis, and this, again, we're just kind of lumping into mitosis, even though they're technically separate. But cytokinesis is the division of cytoplasm into two individual cells. Depending on what our cell type is, cytokinesis is going to happen in different ways. So for our plant cells, we are going to have a cell plate that forms midway between our divided nuclei and gradually becomes the cell membrane. So for our cell plate, when our cells are dividing, if remember, plant cells are usually square, we're going to have this rough plate that forms down the middle that will eventually become 2 cells. In our animal cells, it works a little different, and we're going to form a cleavage furrow that pinches in the cell to create 2 equal portions. And so, yes, we know what we're thinking here, cleavage furrow. So animal cells are round. They're going to separating out to 2 sides. This is going to start pinching in. That looks like cleavage. It looks like breasts. And then it'll eventually become 2 separate cells. So this pinching in structure here is our cleavage furrow. So now that mitosis and cytokinesis is complete, we have created 2 identical body cells. And we're saying they're body cells because mitosis makes all of the cells that make up all the parts of our body. So something to ask ourselves is, how often do our cells divide? Why does this matter? So every cell, depending on what cell type it is going to divide at a different rate based on its own needs. So some examples of this, we have the internal linings of the intestines is every 5 days. So kind of like our cheek cells, where we have saliva, there's stomach acid. So it has to reproduce a lot because we're breaking those cells down as we use them. Then our skin cells every 2 weeks. Again, they're dead. We're flaking them off all the time. Red blood cells every 4 months, and then liver cells a year. So just depending on the cell, it's going to have a different time rate. And then why is it that our cells divide? I'm done growing, but I still need more cells. So we go through cell division for both growth and repair. I'm done growing, but I'm still growing more cells like my intestine cells or my skin cells, and I'm repairing old or worn out cells. And again, I'm done growing, but from my infant, if we remember our first slide with the person that you had to guess, we went from a little fetus to a baby to our angel. And so cells can't just get bigger to grow either. They have to stay small in order to maintain their high surface area to volume ratio. You are not just one giant cell, you're a whole bunch of little tiny cells. So having all of these little cells is the most efficient energy-wise that allows substances to move in and out of our cells more easily. If you were one giant cell, there would be very complex and a whole lot of things going on for one cell. All right, so now that we are done with these notes, if you are listening, I would like you to come tell Mrs. Billups that you heard me say something about the star on the slide. And thank you for listening.